Perfect. All right. We are live. We're broadcasting on Facebook.
Then my name is Jeff Champlin. And with especially Kenneth and Pat, it's our honor tonight to welcome Dr. Aswanth Reddy. This is the last in our series after the apocalypse. And you can see that Dr. Reddy is going to talk with us about the virus and the vaccine. And this is intended in a way, our little contribution to building confidence, which is how in the world we were able to accomplish the miracle that we did in producing the vaccine in less than a year. Or actually, really in about 8 months, come to think of it, 8 or 9 months. Dr. Reddy comes to us, his doctorate in India, would I be right saying Chengalpattu?
Yeah, close.
Close, okay.
Yeah.
It looked like Bengal, so I was sort of going with that.
Yeah, Chengalpattu, yeah. Right, you're right.
And then his residency at St. Vincent Hospital in Worcester, Mass. And I used to live in Connecticut, so I know that's Worcester, not Worcester. So I do know something. He went on to be a chief medical resident there, and then fellowship in hematology and oncology from the University of Connecticut Health, where they also play basketball, especially women's basketball. And he was an integral part there of thoracic oncology, primary research characterizing circulating tumor cells in breast cancer patients. So he brings a distinguished background to us, and we're grateful that he's here. He's married to Mercy Fort Smith cardiologist, Dr. Aswini Kumar, and they have a little baby girl who I don't think is attending tonight.
You may hear some baby noises if my wife signs in. So let's see. Yeah.
Very good. And we're just honored and excited. As I say, this is our contribution to public health and good citizenship in America. And Dr. Reddy, we're all ears.
Okay, thank you. Thanks for your kind introduction. So I tried to make this talk kind of simple, so that everyone takes home something from today's talk and can understand the basics of the virus and how we got into this pandemic and how the vaccine came out within a year. So let's move on. So this is my objectives for today. I'm going to talk about recent advances in understanding viruses, because that's actually related to how they rapidly created the vaccine. And then the next will be about the process of the vaccine creation. And then the last one would be, where do we take this technology from here, and can we use it in other medical specialties like cancer and other therapies? So start with a quick joke there. I hope everybody has no trouble in putting their Zoom in mute. And so a little introduction about viruses. Viruses basically there is a huge debate which goes on, people talk whether viruses are living or non-living. So general definition for viruses, they are very small. They have a genetic material in them, and they have a protein coat in them. And that's pretty much it. That consists of a virus. And they used to classify viruses into different types. So you can see that they used to classify them with regards to shape. They can be of different shapes, and they used to group these viruses with their shapes. Then they classified them in how they live. They can live in animals, they can live in just the plants, and they can actually live in bacteria. They can take up the bacteria, they can grow into the bacteria, and then they can leave the bacteria. And those viruses are called bacteriophages. And then scientists looked closely into the genetic material. Before they did research on this genetic material, they thought all viruses had the same genetic code, basically. But then they realized that they have two types of genetic codes. One is an RNA or a DNA. So RNAs and DNAs are different. They have different sugar structures in them, and they have different molecules in them. I will explain a little bit about it in like the further slides. But you can classify, for example, a virus can be spherical, which we know, which is a coronavirus. I'm sure everybody has seen the structure with the spikes in them. That's a spherical virus. It actually lives in animals, and then it's an RNA virus. It's not a DNA virus. So now the general classification is grouping all these together and then define a virus. And then they are very diverse. And then there are some common features for viruses. They are very, very small, as I said. They should be less than 200 nanometers. They cannot replicate on its own. For example, they can live on the surface. They can live on, like outside environment, but they cannot replicate. They need a living cell for them to replicate. I told you they have the genetic material in them, but they don't have something called ribosomes. Ribosomes are nothing but they are like an instrument which makes proteins, and every living cell has ribosomes in them. And without these ribosomes, the viruses cannot make proteins, so they cannot divide. So they need another living cell to actually replicate and then transfer from one organism to other. So there is a huge debate goes on whether to ask whether the virus is a living thing. The virus does not have ribosomes, I told you, and they cannot make proteins. And they don't have ATP. ATP is nothing but powerhouse. Every cell needs to make its own power for their own energy, and the viruses don't have ATP, so they don't have any energy in them. And so the general consensus is the viruses are basically a parasite, and by strict definition, they are non-living. But there's a huge debate going on between them, because even though they're not living, they can live when they go into another organism. So origin of viruses, how did viruses come into this world? So again, there is no clear explanation on that. There are several theories. The three main theories which is adapted kind of worldwide is, one is they think that they were actually just genetic materials and they were just living on their own. And then they learned somehow to move between one cell to another, and then they cause infections, and then they cause diseases. And that theory is called progressive theory. And the other one is they actually were living organisms before, and through the evolution, they lost all that. I explained about the ATP, ribosomes, they kind of lost all that. Basically, they got lazy. And then what they did was they said, okay, why don't we have to do all this if we can find a host to do all this? So that theory is called a regressive theory. So they were living and then they went back. And the third one, they actually think that these are viruses were existing even before the evolution of life. And then they coexisted with life once the life existed. So that theory is called a virus first theory. So these three theories are mostly accepted, but no one of them is widely accepted. So each scientist differs on their explanations. This is a picture actually showing SARS-CoV-2 virus. This is actually a microscopic picture of the real virus. It's an RNA virus, as I said. This is the one which causes COVID-19 infection. And the size of this virus is about 65 to 125 nanometers. And you can see they are spherical in shape. They actually have these spikes around them. And that's the name. And that's where they get the name from. Coronavirus means crown. And if you look under a real electron microscope, they actually look nice and crown in shape. And that's where they group these viruses as coronaviruses. And how did we get them? So I don't know, most of you would remember the SARS-1, which happened around like 2003, 2004. And this coronavirus 2, SARS-CoV-2, is actually very similar to SARS-CoV-1 in their structure. Most coronaviruses are seen in animals. They don't kill them. They actually live on the animals. They don't infect the animals, but they just replicate. So when you get into very close contact with wild animals, that's when you get viruses. And the most widely accepted theory, or like the scientists believe, is it came from bats, either to close contact or by eating wild animals. And recently, WHO was there in China, and they were investigating this, and they came into a conclusion that the government in China were having these farms for wild animals, and they will grow these wild animals and farms, and they will sell to the people. These farms were developed just to prevent any illegal trafficking of this wild animals, and all these farms were closed in February 2020, and there was no explanation why they closed it. So WHO strongly believes that one of these farms is where the outbreak happened, and these farms were located in the Wuhan province. So that's where they believe it all started. So it's, as I said, the viruses, they use other wild animals to live on them, but they don't really infect them. But when they move to the human host, they infect and then they spread. This is just a table comparing the other coronaviruses. We all know COVID-19. This is a data from somewhere in March. So that's why the cases are 100,000. They just wanted to compare this. The SARS, which we had in 2003, the total were only 8,000 cases, but the mortality rate was very high. You can see 9.6%. And the next one is MERS virus. This was an outbreak in Middle East, Saudi Arabia, primarily. They think this virus originated from camels, the MERS virus, and there were only about 2,500 cases. And you can see the mortality rate is very, very high. It's 34% mortality rate. And then the last one, they compared it with swine flu. You can see the number of cases and the mortality rate is very low. So what this table tells us is an ideal virus should spread very fast from one species to other. I mean, one human being to other. It should not kill the person which is infecting. So in that way, it can spread more. And if it does that, it can actually like infect a lot of people. That's actually, they say, a success of a virus. If a virus kills a lot of people, like how it did, and quickly how it did in SARS and the SARS-1 and MERS, it's actually easy to eradicate that virus. If it goes the other way, it's really gets tough to eradicate that virus. So the mortality rate was 3.4% when we started, but we were missing a lot of asymptomatic cases, and we were not testing enough to identify all the cases. So the current mortality rate is it's approximately 1.8%. So this is the number of deaths in the United States with COVID-19. We started with 1 death in March of 2020, and then 100,000 deaths within 50 days, and then close to like 178 days, we had 250,000 deaths, and then now we have crossed 500,000 deaths. I just wanted to show this picture to see if anybody know what this is. So it may be a difficult picture to explain. So if you look closer, you can actually like kind of guess what this is. This is like the oldest method of genetics, how scientists were detecting genetic abnormalities in people. They actually took a cell, they opened the cell in the microscope, they opened the nucleus of the cell and took out the chromosomes. It's actually the chromosomes. They marked the chromosomes and they marked them with like a blueprint. They have a blueprint, and they will compare the blueprint with this patient's chromosomes, and they will arrange it in a way in numbers. So 1 is chromosome 1, and then you arrange it up to 22, and then XY is the sex chromosome. So this is the oldest method of identifying genetic diseases. So if you look closer in this, so this is actually a normal male karyotype. That's what we call it as, a normal male chromosomes. And then if you look closer in this one, this is an abnormal. So if you look pretty closer, you can see in chromosome 13, there's actually an extra copy. And that's what we call it as an extra chromosome 13 or trisomy 13. So if you go back in the 80s and early 90s, they were actually having a normal chromosome, and they will have a patient's chromosome arranged in this way. And then they will compare chromosome to chromosome, and the length of the chromosome, they will measure it. And that's how they will diagnose genetic diseases. This is like the oldest method, one of the oldest methods of identifying genetic diseases. We still use this rarely, but not very common. I'll tell you why. So if you look at the X here, this is the chromosome I showed you in the previous picture. So if you stretch the chromosome, it stretches and stretches and stretches, and then they form something called nucleosomes. And if you stretch them further, that forms a histones. And if you stretch them further, they're nothing but a DNA, and then the DNA has these proteins in them, or they call it as base pairs. And each set of these proteins forms each gene. So basically, you take a cell, you open the cell, take a chromosome, like these are all the small chromosomes, you stretch them and stretch them and stretch them, all you get is just the base pairs. And the base pairs are pretty simple. It's either A, T, C, G. That's all. It's just these four compounds keeps coming again and again. But when they form a set of compounds, for example, if you take it from here to here, if it's like G, C, T, G, T, C, A, and then the opposite of them, A always pairs with T, G always pairs with C, and then a length of a base pair actually has a function. And if the length of a base pair gets a function, that's called a gene, basically. And what does genes do? Genes actually makes proteins. So again, showing kind of maximize structure of a DNA. I told you base pairs, A always pairs with T, and the side one is actually a sugar and a phosphorus structure. So this is actually a zoomed in version of a gene. So I told you when you take a chromosome and stretch it out, and then each segment of a chromosome where you form a gene has a certain function. For example, when you have a problem in chromosomes, you go in and look for where the exact problem is. And if you see the problem here in chromosome 17, and then if they see that, okay, the base is not pairing that normally. So I told you A pairs with T and then G pairs with C, and they will see, okay, A is pairing with C, which is abnormal. And then they see which is the site of the gene where it's abnormal, and they can identify the gene which is abnormal. And in this case, BRCA1 gene, it's in chromosome 17. It's always in chromosome 17. And BRCA2 gene, it's always in chromosome 13. If they identify this exact location in the chromosome, this exact location and this exact place where it's abnormal, and they can say, okay, BRCA1 gene is abnormal in this patient, and this increases the risk for breast cancer, ovarian cancer. And that's how we study genetics these days. And what does the DNA do? So DNA, it's in the cell, right? DNA, what it does is it actually gives a signal to something called mRNA. DNA forms these small structures called mRNA in the nucleus of the cell. mRNAs, as I said, they're a little bit different from DNAs. And what this mRNA does is it goes from inside the nucleus and it goes to the outside the nucleus, still inside the cell. And what does mRNA does is it goes to the ribosomes. I kind of talked about ribosomes initially. And ribosomes, I said, they are the structures which make protein. And DNA gives a signal to mRNA, mRNA gives a signal to ribosomes, and ribosomes makes protein. This is a basic structure of any gene, any DNA and RNA. So this is like a cell, and the purple one is the nucleus, the DNA. The process is called transcription. It gives a signal to the mRNA, and the mRNA is like a thread. The mRNA moves from nucleus and then goes to ribosomes, and that process is called translation, and ribosomes make proteins. So our body is so to make it very simple, our human body functions with just proteins, basically. And it needs like sugars and other energy, phosphorus and everything, but proteins are the main component of our human living system. And these proteins, each DNA will send a specific mRNA for making a specific protein. And that mRNA will give a signal only to make that one protein. If a DNA is abnormal, for example, if a DNA, as I said, gets a mutation or a gene is abnormal, it sends a wrong signal. A wrong mRNA is formed. And when a wrong mRNA is formed, it's going to give a wrong protein. And this wrong protein is going to make a cell divide and make a cell abnormal. And basically, the cell can turn into a cancer. So that's kind of a basic explanation how a normal cell can turn into a cancer cell from a mutation in DNA. So we talked about how they used to classify viruses and shapes and where they live and whether what's a genetic material. Now everything is genetics, even with viruses. You can see this is a structure explain like kind of representing a coronavirus. They take the RNA material from them. They can actually like make each different gene in that genetic material. So this is actually the genomic structure of the coronavirus 2. It's actually very, very similar to coronavirus 1. That was the reason they were able to get this genome really quickly, because they already had the coronavirus 1 a genomic structure. There was only subtle difference. If you look closer, they divided very important parts of this genome, and each part produces all these things, and these things are nothing but they are just proteins. So this is the most important protein they identified, and this comes from this part of the genome in the coronavirus DNA material, and they named it as spike because they make the spike protein. This is the protein which is made inside, and then they go into this outside the cell, and they stick as spikes in them. And what does the spikes do? This is the most important thing which is needed for the virus to come and stick to inside of our nose and inside of our lungs to infect us. So they identified that this is one of the most important protein which is needed for the virus to infect the human beings. Okay. So what did they do with this genomic technology? So how did they get into rapid vaccine development? So just a quick video explaining. So just a quick video about RNA viruses. So RNA stands for messenger RNA. I told you it takes the message from the DNA and gives it to the outside the nucleus to make proteins. They explain the same thing. So as I said, human cells, they just split up. They just send out a lot of RNAs to make a lot of proteins. And the part which reads these RNA is called ribosomes. And ribosomes are protein makers. And they make proteins, whatever the RNA says. And the proteins, when you divide the proteins, they're called amino acids. Okay. And the scientist actually can design this mRNA outside a living cell in the lab. And when you inject this mRNA, it can actually be taken into the cell, and our cell thinks that there's a message coming from DNA, but instead we are giving that message from outside, and the cell will start making that protein. And that with the mRNA vaccine, what they did was they actually sent the mRNA for the spike protein. I explained it. The spike is the most important thing which needs to attach the cell. So when they injected it, the mRNA went into the cell. Our cells made the spike proteins, and just the spike protein, not the virus. And when the spike protein was circulating in our blood, our body's immune system, it found it, and then it started making antibodies to fight against the spike protein. So we have a lot of antibodies to the spike protein in our blood when we get the vaccine. And when a real virus comes in, we already have the immunity to fight the virus. So basically, we are tricking our body without a virus to make antibodies with this mRNA technology. We don't use any virus. We don't use any genetic material. Like, people have confusion that, they are injecting DNA. No, they're not actually injecting a DNA. They're injecting an mRNA. The messenger RNA, it's actually a one-way pathway. From DNA, it can come out. It cannot go into your DNA and change your DNA. So this is where everything started. So January of 2020, the complete genetic sequence was shared. And within few months, we had the vaccine, and it was first injected in April in Germany, and then on December 2nd, the vaccine was already approved in the UK. That was the first country to approve the Pfizer vaccine. The BNT162 is the Pfizer vaccine. The name comes from BioNTech company. And this is in the US, same thing, December. You can see there on January 10th, the sequence was released. And then on March 16th, they actually started the trial. When they released the sequence on January 10th, within like 10 to 14 days, they were actually ready with a product. And Moderna, the company was contacting NIH to start the trial. And it took a few months to start the trial, and we got it approved on December 11th. The Pfizer was the first one to get approved, and then within a week, we got the Moderna approved here. So this is the timeline of the vaccine. So the Pfizer trial started a little late because it started in Germany first, because it's a German company. So how do we compare to vaccine manufacturing in the past? You can see polio, first animal studies to like eradication, it took a long time. Typhoid, again, took a short time, but not too long, but it did take some long time. And you can see when we have more and more science and more research, you actually see the swine flu vaccine we got the swine flu vaccine pretty quick when you compare to the other vaccines in the past. And this is the fastest vaccine we have ever gotten. It is because we have the technology. So is this RNA technology new? It's actually not. I pulled out this paper, first known paper published about the RNA technology. Actually, if you go to the year, this was published in 1990. And what do they talk in this is they actually inject RNA and DNA vectors into a muscle, and this muscle did not have a protein. When they injected this RNA and DNA vectors from outside into the muscle actually made the protein, and that's what they proved. So they know that if you inject an RNA, but they were doing along with the DNA at that time, but they do know that if you inject an RNA, you can make protein. And this stayed for at least 2 months. This was animal models, but from 1990, we know that the RNA technology works. So I won't be doing justice to the talk if I don't talk about Dr. Carrico. She is an Hungarian scientist. She was associated with University of Pennsylvania. She is one of the gurus in the messenger RNA technology. So she was studying this technology since 1990, but nobody believed in this technology when she did her studies. So she did not get grants, no funding. She was actually didn't even get support from her colleagues. So and the major problem she had was synthetic RNA, meaning you make an RNA in the lab and give it to patients. It can actually our body can think that it's a foreign object coming inside, and it can mount a severe immune reaction and then can reject it. And that was a major problem. So any RNA made in the lab, it had the problem of entering into the cell. And in 1995, she actually got demoted because she didn't have any other research, and all she had was RNA research, and nobody believed it. They said, okay, this is not going anywhere. She even thought that, she was not good enough. She was almost about to quit. She had some family issues that time, but she still kept going. So basic structure of RNA has this four nucleotides. And then they have this sugar structure and phosphorus structure in them. That's a basic structure of RNA. In synthetic form, one of the structures, to be stable, what happened was the structure was when they entered the cell, they were signaling the immune system, because for this to be stable outside, they have to make some changes in one of the RNA structure. So she and another scientist, Dr. Weissman, they actually tweaked this RNA, and then they made it in the lab to the point that it will not create any immune system. And this RNA is actually called hybrid RNA. When the hybrid RNA is injected into the human body or an animal model, it actually the human or the animal body thinks that it's actually a normal RNA coming, so it doesn't mount an immune response. So these are the two scientists who came up with this technology in actually 2005. So if anybody asks like how we were able to get vaccines so quick, it's because this research was going on, started in 1990s, and 2005 they were able to create a hybrid RNA. She even licensed this technology in 2005. In 2007, actually another scientist from Boston, Derek Rosai, he looked into this technology and he studied this to create stem cells in his lab. He was not actually a vaccine scientist, or he was not like a virus scientist, but he wanted to create stem cells in the lab without using stem cells from embryo to avoid some ethical issues. So he was using this technology to make cells in the lab. And he's actually a co-founder of Moderna. And if you see the name of Moderna, it actually came from modified RNA. And he actually left the company now. He's not with them anymore, but he was one of the co-founders of Moderna. And the other two scientists, Ugur Sahin and Özlem Turek, they were from Germany. They are Turkish scientists, but they moved to they lived in Germany, and then they are husband and wife. They formed a company, and that company is BioNTech. And this company is partnered with Pfizer to make the Pfizer vaccine. And the BioNTech is actually not a virus company, or they are not a vaccine company. They were actually doing cancer immunotherapy research using mRNA technology, and they had all the technology. So they saw this opportunity coming, and then they made the vaccine so quickly. And does the vaccine work? So this is a graph which compares the two vaccines. The purple one, I mean the blue one is actually the Moderna. You can see the graph which goes up. It's actually the graph in which the patients got placebo, and the graph which lies close to the bottom line. It's actually people who got the vaccine. It actually works. You can see the graph actually separates around like 14 days in both the vaccines. That means that it prevents you from getting an infection if you get the vaccine, and the effect starts even after 14 days. But you can see both the lines, they don't touch zero. So that means that you still have some risk of getting the virus even if you get vaccinated, but the risk is extremely low, which is around less than 5%. And this is a table, just a general table. This is the five vaccines. The second row shows how many patients got the vaccine. And if you see the other columns, it's just zero. What it says is whoever got the vaccine for COVID in all the trials, nobody went to the hospital, nobody died from COVID, nobody had severe disease. So what this tells us is the vaccine, one, it prevents you from getting the infection. Second, it actually prevents you from getting a severe infection to the point that you don't go to the hospital. You end up most likely with a mild infection. Same concept which we use with flu vaccine. A lot of people are skeptical getting flu vaccine because they'll say you still get a flu, but the reason why we give flu vaccine is when you get it, you already have an immunity. You get a less severe infection with the vaccine. So we talked about the trial data, but what about in real world? This is one of the earliest data which came out from England. You can see these two, when the risk of hospitalization unvaccinated, it was 15%. Within 14 days, it came down to 14%. After 14 days, the risk of hospitalization was 9%. And if you see this, the risk of death actually dropped within 14 days, dropped to 10% from 13.4%, and then dropped to 5.8% after 14 days. So this is one of the first real world data which showed that the vaccine works. This is from the Pfizer vaccine. So do we need masks then? Yeah, for now we need to wear masks. A lot of people ask me, do we have to wear masks because we already got the vaccine? So you got the vaccine, your blood has a lot of antibodies to fight the spike protein from the virus, but the virus can actually replicate in our nose and the back of our nose. So you have to create a local immunity to get rid of the virus from the nose or the nasal epithelium. So you won't get an infection, but the virus can linger in your nose and you can still keep spreading if you don't wear a mask. And that's the reason if you're coming into contact in big groups or if you're coming into contact with unvaccinated people, it's better to wear a mask. And this is the reason why, because you can still have viruses, circulating from your nose. You can ask what can be done for this. They're looking into nasal sprays. I'm more excited about this because when you give a virus nasal spray, there used to be there still is. There is a flu vaccine which is actually given as a nasal spray, which is a live virus. That's why we don't use it widely. So when you use it as a nasal spray, it actually creates a local immunity. So you can be able to get rid of the virus even before it enters your nasal epithelium. So these are the studies ongoing. And there are a lot of people who are afraid to get a shot. So there are other ways like a patch, an implant, and even a sublingual tablet. They are looking into making vaccines by this route. But I'm more excited into this technology, like a nasal spray, just 2 sprays in your nose, and then you're vaccinated. And so we talked about a little bit about wearing a mask. Is there any other benefits of wearing a mask? Actually, yes. So if you see this graph, this is actually number of flu cases, which comes every year. So we say the flu season starts like in winter, goes up, peaks in February, and then comes down March, April. So this is the curve. This is a graph which they published around January time. You can see the red line is actually for this year, 2020, 2021. We actually had extremely low flu cases this year. When you compare the graph, the red graph should have been here somewhere. One reason they're saying is we are not like getting into big groups. The other main reason they're saying is actually from wearing a mask. So there are some more other benefits of wearing a mask. And so why is there so much skepticism about vaccine? I thought about it a lot, and I think I came to this conclusion. The growth of social media, I think there were a lot of information spreading everywhere. And if you see this graph, this is just a joke, but you see it actually correlates. If the growth of social media all through the years, and it actually correlates with with how much information people get and how much people are skeptical, and they're worried because there's so much fear which is going around just from stuff you read in social media and all of the stuff. So we now talked about what does a virus look like, how we can genetically code a virus, and how we were able to make a vaccine so quick, because we had this technology all this while and we were not using that much, mainly because of funding. And because of COVID, all these companies got a huge funding. So and the government said, spend all the money, do the research. If the research fails, it's okay, we will eat up the cost. And that was the main one of the main reasons the scientists say we were able to get the vaccines at this rapid pace. One is we had a technology, the second one is funding. So what do we use this technology from here in other infectious diseases? So I pulled out a table from a review table in which the RNA technologies, all these are mRNA technologies, messenger RNA technologies, and they use it for HIV, rabies virus, a lot of HIV clinical trials going on, a Zika virus, and an influenza virus too. So we will be seeing more and more of these vaccines coming out in the near future because we know it works, and companies, again, a lot of companies are ready to fund this research now. So I was interested, when all these trials started, there were a lot of trials going on, and some say it's already completed. Nobody knew about mRNA vaccines or technology before Pfizer or Moderna vaccines. So I was interested to see when all these trials started. The first trial, this one, the Ergos Therapeutics, it's actually started in 2008. So basically what I learned is this is not a new technology. The research has been going on. I listened to one of the HIV scientists talking about this. The main reason these studies were not able to be completed and the vaccines are not produced, it's again, one thing, a lot of people did not believe in this technology. Another thing was funding. Big companies were not ready to fund the vaccine. So they always, so you would have heard that a vaccine takes like 10 years to come out. That's because they want to do the phase 1 trial first, see what dose is good in phase 1, then they move on to phase 2, which is a safety trial. See, they will give it to a bunch of people and see if it works. Then they will move on to phase 3, and then comes to the market. So in this, they actually excluded most of the animal studies in COVID vaccines. They combined phase 1 and 2 together, which we do it in some of the cancer trials. and then they moved on to phase 3 really quickly when they got interim results. So that was another reason we were able to get the vaccine ready by end of within a year. So RNA technology in gene therapy. What is gene therapy? Gene therapy, this is a simple chart which explains what is gene therapy. Gene therapy is used when you have an abnormal gene. Basically, you have your DNA has an abnormal gene, you're born with that abnormal gene. Because of that, you have a disease. So what they do is most of the cells start from the stem cell. Right? So if you change the stem cells, you can actually treat the DNA. So they take the stem cells out, they give patients chemotherapy to clean up all the stem cells left in the body. They actually use a virus vector. You can see this is a virus. Mostly they use a virus called adenovirus, and they use this DNA material, and they can inject the normal DNA material in the virus. And what this virus does is, this is an inactivated virus. They cannot cause infection. It just takes the genetic material, goes inside the cell. This is all done in the lab. They put both of them in a culture where this genetic material is transferred into a stem cell, and then the stem cell repairs itself, becomes like a normal stem cell, and then you give it back to the patient as like a fresh new stem cell, and then the disease is treated. This is a gene therapy. There's actually gene therapy going on for a lot of diseases. Most of the diseases have abnormal gene and abnormal protein. One of the diseases I was interested in, sickle cell disease. We had a big sickle cell population in University of Connecticut, and there's already a first patient who already got the gene therapy for sickle cell disease, and she's doing well so far. I checked it yesterday, and she's doing well so far. So we'll see more and more gene therapies for sickle cell disease in the near future. So you use a DNA here. If you see, they use the virus, they use a DNA material into the virus, give it to the cell. It's a cumbersome process when they do it. Why don't they do it directly into the patient with an mRNA technology? And that's what they're looking into it. So just take the mRNA. Don't worry about the DNA. Give into the patient's bone marrow, and then create a new stem cell inside the patient, so that you can avoid the process of giving chemotherapy, getting the cells out, giving it back to that. And that technology is under research now, and they are looking into a lot of gene therapies. They're trying to transition from a DNA-based virus vector-based technology into an RNA-based technology. And what about in cancer? So so far we talked about abnormal gene, and abnormal gene making an abnormal gene is not making a protein, or it can make a bad protein. In cancers, the problem is making a lot of bad proteins, which makes the cell keep growing, divide, and then it can spread. So that's a problem in cancer. So there are RNA technologies in research, but these RNA technologies, they don't use it to stop a protein or make a protein. Why? A cancer cell does not have one gene problem. That's a problem. That's the characteristic of a cancer cell. If you break a cancer cell's nucleus, and then if you read the genetic code in a cancer cell, there are multiple mutations going on at the same time. There is no cancer cell with one mutation, but one is always predominant. So that's what we call it as a driving mutation. So if you block the driving mutation, another mutation will come in, and then that will start making the cancer grow. So that's why the targeted therapy we use, it will block one of the gene drivers, and for some time the patient will be responding, will be doing good, and then all of a sudden we'll see that the cancer is growing. I'm like, why do you? And we'll check that they still have the mutation, but they will have a different gene which kind of overtook, and then it's growing. So it's always a problem to stop a gene from in a cancer cell. So instead, what they did was they took the cancer cells, they took patients' immune cells. We all have immune cells, and cancer cell is a bad cell. So the immune cells should kill the cancer cell, right? But the cancer cells are avoiding our own immune system, and they're hiding. So what they did is they took the immune cells, they took the cancer cells, they took some proteins from the cancer cells, and they formed something called neopeptides, which just means small proteins. And then they used these proteins to activate our immune cells. They said, okay, they made our immune cells to identify these abnormal proteins. And then they activated these dendritic cells. Dendritic cells are a form of immune cells which will go and kill one of the cancer cells. And then when you cultivate these immune cells in the lab and you multiply them into like millions and millions of copies after activation, you give it back to the patient. So basically, you're taking only few cells, activating our own cancer cells to activating our own immune cells to identify the cancer proteins, and then multiplying these activated cells into like millions and millions of copies, giving back to the patient. So this is the concept which is called cancer vaccine, and this technology is highly dependent on RNA technology. So we talked about there are some conditions in which there is an excess protein. It's not a cancer, but people can have excess protein in the body, right? And that can happen from a mutation. And can we turn off this excess protein with an RNA technology? Actually, yes. So this is actually an FDA-approved therapy. This is a pretty rare condition. It's called a TTR amyloidosis. I'll explain it to you in a simple terms. Amyloidosis is abnormal protein. In this condition, the liver makes a lot of abnormal protein by a mutation. It just keeps making it. And these abnormal proteins circulate in the blood, and then they can go sit in the heart. They can go sit in your nerves and cause a lot of trouble. And what they did is, how can we silence this protein? So what they made is something called silencing RNA. So and this is a derivative of a mRNA. So what they do is they inject this silencing RNA. It's called siRNA. They go in and what they do is they go into the cell, same technology. The mRNA, I told you mRNA makes proteins to ribosomes. The silencing RNA competes with the mRNA, the abnormal mRNA, and then they go and attach to the ribosomes, and then they don't make any protein. So that's basically they silence the ribosomes and do not make any abnormal protein. So when they go in, this drug goes and silences the cells, less protein is secreted because the ribosomes are all occupied by the silencing RNA, and then mRNA cannot make more protein. So less proteins, less amyloid deposits, and patients actually saw a response. This drug is actually approved for neuropathy. This condition, the TTR amyloidosis, which is abnormal protein, causes severe peripheral neuropathy, meaning damage to the nerves. When they gave this drug, it reduced the proteins in the blood, and the patient had a clinical response. I was actually this got FDA approved around 2018. I actually was writing a book chapter with one of my mentors in Connecticut, and that's when we came across this drug, and it's actually got FDA approved now. It's used for peripheral neuropathy from this condition. So basically, you can tell a cell, any cell, to make a new protein, or you can actually send a different RNA to silence and then not make a protein. It gets tricky when you deal with a cancer cell. I told you, because there are a lot of proteins going on at the same time. One is predominant at one point, you silence that, you can have another one coming out. That's why instead of focusing on blocking the proteins in a cancer cell, they are focusing research on tricking our own body's immune system to fight against the cancer using the same mRNA technology. So that's why the mRNA technology per se, it's not directly helpful for cancer patients, but indirectly, tricking the immune cells, it's actually very successful. So there are a lot of trials going on. The picture I showed you, it's actually from and this is actually from a melanoma clinical trial, which is ongoing. So they take the cells from the patients, send it to a lab where they can do all this, and then they make like a bag of cells. They give it back to the patient, and then we inject that to the patient. So this is ongoing. And I know one of the researcher in University of Connecticut, he's actually working on very similar concept, but in ovarian cancer. He's been doing this research for almost 20 years. What he does is he takes the patient's cancer. Actually, part of the tumor specimen goes to Germany, where they identify these proteins. And when they identify these proteins, they form they attach these proteins to the immune cells and see which immune cells captures them. And if any of the immune cells captures them, they select those immune cells and they multiply them into million more, and then give it back to the patient. Yeah. So that's kind of the technology. They look into a lot of cancers now. So instead of giving chemotherapy and killing the cancer cells, because you don't specify which cell to kill, right? Chemotherapy is a generalized treatment. You give it to the whole body. The focus is more on like personalized care. Each individual patient will have a different cocktail in future. That's what I'm seeing in future. You go in, you get a biopsy. They'll say, okay, we'll go to the lab, and you will hear from us in like several weeks, and then you will get your own personalized cocktail, and that will be the treatment for you. And that's what the future I would like to see too, rather than giving a standardized treatment to all the patients. And that's my overview about viruses and vaccines. And thank you all for listening. And if you have any questions, I'm happy to answer.